Good morning students, we will continue topic chemistry of protein competition number 5.1 and today's subtopic is different levels of protein structure including disulfide and weak bonds with examples and its clinical significance. Protein structure can be studied at 4 different levels. First is primary structure, secondary structure, tertiary structure and quaternary structures. So we will see one by one. Before studying the protein structure, we should know the bonds which are present in these protein structures, and these are the various types of bonds which stabilizes the various protein structures. And these bonds are peptide bonds, hydrogen bond, disulfide bond, electrostatic bond, hydrophobic bond and van der Waals forces. Peptide bond we have already covered in the last lecture, but still it is an overview. Peptide bond is formed between the carboxyl group of previous amino acid and amino group of next amino acid by dehydration. So COOH group and NH2 group condenses with dehydration to form CONH group or peptide bond. Next bond is very important bond, that is hydrogen bond. It is formed between the oxygen atom which is attached to the carboxyl carbon involved in peptide bond and hydrogen atom which is attached to the amide group involved in the peptide bond. So it is formed between CO and NH group of two peptide bonds, not in the same peptide bond. So this is the diagrammatic representation. Hydrogen bond in alpha helix structure of protein. Here if you see, hydrogen bond is formed between CO and NH group of two different peptide bonds. In alpha helix structure, these peptide bonds are formed between the first amino acid and the fourth amino acid. Alpha helix structure of protein is the secondary structure. In the secondary structure of protein, the amino acids will come little close to each other, therefore it is easy to form a peptide bond between first and fourth amino acid. So this is the diagrammatic representation of alpha helix structure of protein, where you can see the hydrogen bonds shown by dotted lines between CO and NH of first and fourth peptide bond. So same, it is formed, hydrogen bond is formed in, now we will see hydrogen bond in beta pleated sheet structure. In beta pleated sheet structure, hydrogen bond is formed between, again same, oxygen atom which is attached to the carboxyl group involved in the peptide bond of one chain and hydrogen atom which is attached to the amide group involved in the peptide bond of other chain. So here the formation of hydrogen bond is between the same groups, that is CO group and NH group, but in between two different chains. So by this figure you can understood, here two different zigzag type of polypeptide chains are kept side by side. This is called as beta pleated sheet structure. If you see a dotted line between O and H atom of two different peptide bonds of two different polypeptide chains. So same is shown here. Now disulfide bond. Now this disulfide bond is formed between sulfur atom. Therefore it is called as disulfide bond, and this sulfur atom belongs to a amino acid cysteine. So basically it is formed between two cysteine residues or two cysteine amino acids of one or different polypeptide chains. If disulfide bond is formed within a same polypeptide chain, then it is called as intra-chain disulfide bond. In this case, disulfide bond is formed between two different polypeptide chains, so it is called as interchain disulfide bond. Next bond is electrostatic bond. These are attractive forces between opposite charges or repulsion between two similar charges. So electrostatic bonds are both attractive as well as repulsive forces. Example, negative charges produced by acidic amino acid like carboxyl group of aspartic acid and glutamic acid, and positive charges produced by basic amino acid like amino group of lysine and arginine. So these are the amino acid which produces charges in a polypeptide chain. Now you see this. Here two polypeptide chains are there. In the first polypeptide chain, aspartic amino acid is shown which has carboxyl group with negative charge, and in the lower polypeptide chain on the left side, arginine amino acid is there which have amino group with positive charge. So these positive negative charges have attraction between them and have attractive type of electrostatic bond. In the next, lysine amino acid is shown in the above polypeptide chain with amino group and positive charge, and in the lower polypeptide chain, glutamic acid with negative charge on carboxyl group is shown, and again positive negative get attracted, and it is an example of attractive type of electrostatic bond. But sometimes instead of oppositely charged groups coming together, similar charged groups also came together, then they get repulsion from each other, and they are also included in the electrostatic bond. Next bond is hydrophobic bond. These are formed between two polypeptide chains or two non-polar hydrophobic side chains of a polypeptide chains. Again, it is shown below. In the above polypeptide chain, the alanine amino acid is there. The side chain present in alanine is CH3 group. In the lower polypeptide chain, valine amino acid is shown. Valine is a example of branched chain amino acid. In the side chain of valine, we have CH3 group. So all these groups are hydrophobic in nature, and when such groups came close to each other, they have some attractive forces between them, which is called as hydrophobic bond, though they are very weak bonds. And the last type of bond is van der Waals forces. These are attractive forces between all atoms due to oscillating dipoles. These are weak forces, but they are present in very large number. These forces preserve the non-polar interior structure of proteins. When proteins are folded, then the non-polar part of the protein hide inside, and the polar part of the proteins get exposed to the surface. So van der Waals forces helps in preserving the interior non-polar structure of the protein. After completing the forces, we will start with a structure of proteins. Whenever there is a question on structure of protein, you have to describe about these bonds. So first structure of protein is primary structure of protein. Primary structure of protein denotes the number and sequence of amino acid in a protein joined together by peptide bond. That means it is nothing but a straight chain structure of a protein is called as primary structure of protein, which is shown here. Here peptide bond joins various amino acid while side chains remains outside. So in this case, peptide bond forms the backbone of the polypeptide chain. Something importance about primary structure. Primary structure gives us sequence and number of amino acid in a protein. Now why this sequence and number of amino acid is important? When the sequence of amino acid changes in a polypeptide chain, it becomes a different protein. Or if the number of amino acid changes in a polypeptide chain, it becomes a different protein. For example, here I have given examples of 3 proteins. In protein 1 and protein 2, the amino acids are same, that is methionine, threonine, valine in first protein, and in second protein methionine, valine, threonine. The amino acids are same, but sequence of amino acid is different. Now if you compare protein 2 with protein 3, then we see that the sequence of amino acid up to third amino acid is same, but extra amino acid glycine is present in protein 3. So number of amino acid in protein 3 is more as compared to protein 2. Therefore protein 3 is different from protein 2. So if the sequence of amino acid changes, new protein is formed. If the number of amino acid changes, new proteins are formed. And all these proteins have different properties and different functions. So when the primary structure of protein changes, that means sequence or number of amino acid changes, the property and function of protein also changes. That we can study by example of sickle cell hemoglobin, where only at 6th position of beta globin chain glutamic acid is replaced by valine, that is sequence of amino acid changes. Therefore the property and function of hemoglobin S is different from normal hemoglobin A. Forces present in a primary structure of protein are peptide bonds and disulfide bonds. Now importance of primary structure is that primary structure decides the three-dimensional structure or tertiary structure of the protein, which in turn decides the function of proteins. A change in single amino acid of protein leads to loss of structure and function of proteins. Again, it is studied with the help of example taking hemoglobin S, where substitution of one amino acid leads to different structure as well as altered function of hemoglobin S. Primary structure helps in determining the sequence of amino acid in protein. If you want to study the sequence of amino acid in a particular protein, then you have to convert that protein into primary structure, then you can find out its sequence. Also primary structure helps to count the amino acid in a protein or numbering of amino acid in a proteins. So this is the importance of primary structure. So this is the example proinsulin, where peptide bond and disulfide bonds are present. This is a mature insulin with two polypeptide chains lie side by side in a straight chain manner joined by disulfide bond. Now secondary structure of protein. Secondary structure denotes the steric relationship between amino acids which are located relatively close to each other in a polypeptide chain. So this is the definition. Secondary structure is formed by folding or coiling of polypeptide chain in the form of helically coiled or zigzag linear pattern. Two types of secondary structure of proteins are seen in our body: alpha helix and beta pleated sheet structure. So this is an example of alpha helix structure of protein. Here polypeptide chain is coiled around the longitudinal axis to form a right-handed helix. Each turn is formed by 3.6 residues. Here residues means amino acids. Pitch, that is distance between two equivalent points on turn, is 5.4, and the distance between two successive amino acid is 1.5 angstrom. The polypeptide bond forms the backbone and side chains of amino acid are extended outward as shown in figure. The coils are stabilized by hydrogen bonds formed between carbonyl oxygen of first peptide bond and amide hydrogen atom of fourth peptide bond, that we have already studied in hydrogen bonds. Left-handed helix is rare because amino acids found in protein are of L variety or L amino acids, which does not suit for left-handed alpha helix structure. Bonds present in alpha helix structure of proteins are peptide bonds, disulfide bond, hydrogen bonds, electrostatic bonds, hydrophobic bonds, and van der Waals forces. Now, some amino acids are compatible with alpha helix structure and some amino acids are non-compatible with alpha helical structure. That means in that structure they are not suitable. For example, proline structure of proline amino acid is cyclic. That means R group is present in the cyclic form, and this is not compatible with right-handed alpha helical structure. It alters the function of proteins if it is present. Therefore, very few proline amino acids are present in alpha helical structure. Apart from proline, valine, leucine, isoleucine, these are branched chain amino acids which are shown in lower figure. So side chain consisting of large branches. So side chains are bulky in this case, and these side chains are too large to be accommodated in the alpha helix structure in large quantity. Hence, these amino acids are also found in less number in alpha helical structure. Next, amino acids which are not compatible with alpha helix structure are charged amino acids like glutamic acid, aspartic acid having negative charge, lysine and arginine having positive charge. So, because of presence of these charges in the protein molecule, there is attractive or repulsive forces generated, and which also destabilizes the alpha helical structure, and hence such amino acids are also present in less quantity in alpha helical structure. Up till now we have studied the non-compatible amino acid. Now we will study the examples of the amino acids which are compatible with alpha helical structure, and these are alanine, glycine and phenylalanine. They are most common amino acid found in polypeptide chain of alpha helix because they are small and uncharged. Hence they can easily accommodate in a alpha helical structure without disturbing the structure. So these amino acids are found in large number in alpha helical structure of proteins. Now examples of alpha helical structure. Alpha helical structure of protein is seen in globin chain of hemoglobin and globin chain of myoglobin. Myoglobin is a protein present in muscles. The function of myoglobin is to store the oxygen, whereas the function of hemoglobin is to transport the oxygen. Now this alpha helical structure of protein is also found in alpha keratin, protein present in nails, hairs and skin. After alpha helical structure, we will study beta pleated sheet structure. The polypeptide chains here are fully extended into zigzag manner. The distance between two adjacent amino acids is 3.5 angstrom. It resembles like series of pleats and hence called pleated sheet structure. This is also example of beta pleated sheet structure. If the C terminal and N terminal of two adjacent polypeptide chains lie opposite to each other, then they are called as antiparallel strands. And if C terminal and N terminal of two adjacent polypeptide chain lies on same side, then they are called as parallel strands of beta pleated sheet structure. Now these are the bonds present in beta pleated sheet structure of protein: peptide bond, disulfide bond, hydrogen bonds, electrostatic bond, hydrophobic bonds and van der Waals forces. We have already described these bonds in details previously. Now we will see the amino acids found in beta pleated sheet structure. Glycine, serine, alanine are the most common amino acids found in beta pleated sheet structure. That means they are compatible. Whereas proline, again, found in small quantity in beta pleated sheet structure, because if proline is present in large quantity, it causes kink in the polypeptide chain. So this proline is not compatible with beta pleated sheet structure. Example of beta pleated sheet structure is flavodoxin, skilfibroin and carbonic anhydrase enzyme. Now tertiary structure of proteins. The polypeptide chain with secondary structure may further folded and superfolded and twisted about itself in an aqueous medium to form a spherical or globular shape is called as tertiary structure of protein. That means secondary structure is superfolded so that it can accommodate in small space. Now such structure is called as tertiary structure of protein. Now by definition, tertiary structure of protein denotes a steric relation between the amino acids which are located far away from each other in a polypeptide chain but came close to each other by folding and superfolding. So in tertiary structure, the proteins which are far away from each other may come close to each other. It decreases the actual dimension of proteins. The tertiary structure of protein, in tertiary structure, functional groups came close to each other which are situated away from each other in a polypeptide chain. Here I have shown a protein with amino acid A5 means 5th amino acid in a polypeptide chain. A100 means 100th amino acid in a polypeptide chain. A200, A250, A300 and A500 means 500 amino acid. So all these amino acids have came close to each other due to folding and superfolding with their functional groups like SH, OH, carboxyl group, SH again OH and amino group. So functional groups have came close to each other, and when such functional groups came close to each other, they perform function, and hence tertiary structure of protein is called as functional proteins. Why? Because during the tertiary structure formation, all the functional groups of proteins came close to each other due to its folding. During the formation of tertiary structure, the folding and superfolding occur in such a manner that hydrophobic part of side chain of amino acid moves inside or interior, whereas hydrophilic part is present on the outer surface to face the aqueous medium outside. Factors responsible for tertiary structure formation. First is primary structure. I have already told you that primary structure decides the tertiary structure. Next factor is temperature. When the temperature changes, the tertiary structure get disturbed. Next factor is pH. When pH changes, the charges present on the amino acid also changes and they may lose the tertiary structure. Then next factor is chaperones. So all these factors decide the tertiary structure of proteins. Now what is chaperone? Chaperone are also called as polypeptide chain binding proteins. They are specialized proteins found in cells. They assist folding of proteins by recognizing special sequence. So that means chaperone knows where the protein polypeptide chain should fold and in which manner it should fold. So they assist the folding and helps to generate a tertiary structure of protein in a proper manner. Chaperone also increases the rate of folding, so that they are also called as catalyst. Means they make the protein folding faster. When the protein is synthesized during translation, these chaperones will help, or they increases the rate of folding, and therefore they are called as catalyst. They possess ATPase activities. That means they take the energy by breaking the ATP molecules. Many chaperones are also called as heat shock proteins. Defective folding in protein leads to Prion's disease, and it was found that in Prion's disease the function of chaperone is disturbed. Now what is meant by domains in the tertiary structure. Now polypeptide chain is long. See this figure. This is a single polypeptide chain. Now this polypeptide chain is very long. So what will happen? This polypeptide chain folded into 3 or 4 domains. So some part of polypeptide chain folded into tertiary structure which performs some function is called as domain. It is a structural and functional protein. Generally polypeptide chain with 200 or more amino acid consists of 2 or 3 domains, and these are interconnected by polypeptide chain. Each domain work independently. So this is the various domains present in the polypeptide chain. Actually in this way the tertiary structure is present in various domains. The bonds which stabilizes the tertiary structure of proteins are again peptide bond, disulfide bond, hydrogen bond, electrostatic bond, hydrophobic bonds and van der Waals forces. Examples of tertiary structure is seen in various enzymes and structural proteins. Now the amino acids which are compatible with tertiary structure of proteins are again alanine, glycine and phenylalanine. They are the most common amino acid found in polypeptide chain of tertiary structure of proteins because they possess side chain as small and uncharged. Next, quaternary structure of protein. When 2 or more polypeptide chains came together to perform some function, then it is called as quaternary structure of protein. Each polypeptide chain is called as subunit. When these subunits are separated, then protein loses its function. In quaternary structure, it was found that when 2 or more subunits came close to each other, they maintain functional cooperativity between them. So what is meant by functional cooperativity? That we will understand by taking example of hemoglobin. So hemoglobin is made up of 4 polypeptide chains, which is shown by 4 squares. Now on the left upper side I have shown oxygen molecule binds with one polypeptide chain. In this case, in the lungs, when oxygen molecule binds with one polypeptide chain, suddenly the remaining 3 polypeptide chain will show high affinity towards oxygen and binds with the oxygen easily. Similarly, when this hemoglobin goes to the tissue, if one of the polypeptide chain delivers its oxygen to the tissue, then suddenly the remaining 3 polypeptide chain shows decreased affinity towards the oxygen and donate their oxygen to the tissue. This is called as functional cooperativity, and it is seen only in quaternary structure of proteins. The bonds present in quaternary structure of proteins are again peptide bond, disulfide bond, hydrogen bond, electrostatic bond, hydrophobic bonds and van der Waals forces. Example of quaternary structure of protein is hemoglobin, creatine kinase, lactate dehydrogenase, etc. Now, when we study the thermodynamics and stability of various structure of protein, then it was found that formation of secondary, tertiary and quaternary structure of protein follows thermodynamic principle. Energy content decreases from primary to quaternary structure of protein. Therefore, quaternary structure of protein has least energy and most stable structure of proteins. So this is about the various structure of proteins. Now we will study a small topic which is called as denaturation of proteins. When secondary, tertiary and quaternary structure of protein which we have studied that get disturbed, then it is called as denaturation of proteins. It may be reversible or irreversible. In both cases, protein loses its function. So this is the diagrammatic representation where I have shown on the left side a tertiary protein is there, after denaturation it becomes unfolded and its structure becomes like this. Now from this figure what we have understood? From this figure we understood that on denaturation, first thing is that size of protein increases. Second thing is the amino acids which are previously close to each other, now they got separated from each other, and this is the reason why protein loses its function. While describing the tertiary structure, what I have told you, in tertiary structure of protein, the amino acids which are far away from each other will came close to each other with their functional groups, and all these functional groups then perform a function. But during denaturation, all these amino acids go away from each other and protein loses its function. Mechanism of denaturation: unfolding of protein chain due to disruption of many weak bonds which maintain secondary, tertiary and quaternary structure. So bonds becomes disturbed. Primary structure is maintained because peptide bonds are strong and covalent bonds. So during denaturation, primary structure of protein is not disturbed. Now factors which causes denaturation of proteins are change in temperature, change in pH, mechanical, means if you take a protein in a solution and if you shake it vigorously, then protein loses its function. Some organic solvents like alcohol, acetone, acid, alkali, urea, detergent, etc. If you add these to the protein solution, again proteins undergo denaturation. Protein shows denaturation by pressure, ultraviolet light and ultrasound. So these are the factors responsible for denaturation of proteins. Now what happens after denaturation of proteins? First is protein loses its function. Second, protein becomes insoluble. Next, viscosity of the protein solution increases. But advantage is that protein can be easily digested. Why? Because we have seen on denaturation, size of protein increases. When the size of protein increases, there is more area available for the action of digestive enzymes. Therefore, boiling of egg is easily digested, boiled egg, as compared to raw egg. So this is about various structures of proteins and denaturation of proteins. Sometimes it may be asked as a long question or short note, or denaturation of proteins may be asked as a separate short note. Thank you very much.